confirm?
Yes, we have done, but up to fifth point.
Up to fifth point. To add two more points. Okay, so all of you add two more points in mitosis and meiosis. Single karyokinesis in mitosis and two karyokinesis, means two nuclear division in meiosis. Mitosis occur in somatic cells or growth cells of the body, but meiosis division occur in reproductive cells or gametic cells.
Miss, what is the meaning of somatic cells?
Somatic cells means growth cells of the body, which involve in the growth of the body. And gametic cells means which involve in reproduction. Done.
Yes, mister.
Okay. Now move to significance of meiosis. See, these are the significance of meiosis. Meiosis first significance or importance is meiosis allows genetic variation in sexually reproducing organism. In sexually reproducing organism, male and female parents are separate and they produce male and female gamete separately, and both the gametes are fused to form a new organism. So there is genetic variation. So new organism is not the exact copy of their parents. So there is slight variation, and that variation is called genetic variation. Okay, so it occurs only due to meiosis division. In mitosis division, you have written now identical offsprings are daughter cells are produced or offsprings are produced. Identical means exact copy of their parents, and unidentical means they have genetic variation. These genetic changes or genetic variation leads to evolution, and evolution leads to speciation. Speciation means formation of new species. Okay. Evolution means successive change. Okay, over a period of time. So successive, continuous, slow changes, genetic changes is called evolution. And this evolution, this slow changes leads to formation of new species. Like human beings are evolved from monkeys due to genetic changes, due to genetic variation. Monkeys transformed into human being over a long period of time. So monkey to human being is a process of evolution, and monkey is another species, and then new species human being evolved. It provides, third point is, it provides for mixing up of genes, exchange of genetic materials, and this is called crossing over. So exchange of genetic material means maternal and paternal. Maternal means mother and paternal means father. So maternal and paternal chromosomes are get mixed up This is called crossing over. I'll show the diagram. And after crossing over, new combination of genes are produced. That new combination of genes are called recombinants. And the process of recombination, formation of new combination of gene is called recombination. So these are only due to meiosis division, because when gametes are fused together, then maternal and paternal chromosomes are get mixed up and few form recombinant. Fifth one is chromosome number reduced to half. We have seen meiosis division means diploid chromosome undergoes meiosis division to form haploid gametes. So chromosome number reduced to half, so that on fertilization the normal number is restored. So n number, haploid means n, two gametes are fused together, that means n plus 1. So after fertilization, after fusion of gametes, 2n number maintained in the zygote, that is normal number. Understood all of you? All the points are clear?
Yes, miss.
Okay. Now see the diagram of crossing over. What happened actually? There are two homologous chromosomes. Homologous chromosomes means having similar shape and size. I'm writing in the chat box. Homologous chromosomes means chromosomes having similar shape and size, that are called homologous chromosomes. So homologous chromosomes align together, maternal and paternal, and then chromosomes cross over. Maternal and paternal chromosomes cross over. So chromatids cross over. Okay, these two chromatids are crossover and it form X-like structure. This X-like structure is called chiasma. Okay, this is X-like structure formed during crossing over or due to cross of chromatids, sister chromatids. And after that, see in the diagram, genetic materials are exchanged in these two chromatids. So red one is here, a small amount of red one is here, a small amount of blue one is here, small portion of blue. So these two chromatids exchange their genetic material and form new combination. So these two are called recombinant. Okay, these two are called recombinant chromatids which exchange their genetic material, and the chromatids which does not exchange their genetic material are called non-recombinant. Okay, this is the process of crossing over. Okay, so after exchange of genetic materials, few characters of father and few character of mother is transferred, exchange. Clear all of you?
Yes, miss.
Okay. Now see one video which is of mitosis division. Watch the video.
Mitosis. In this module, you will learn about mitosis. Mitosis.
Are you able to hear, children? This is audible?
Yes, miss.
Okay. Okay, see.
Is a process of cell division that makes possible regeneration of body parts. To elaborate, mitosis is the separation of nuclear chromosomes into two identical daughter nuclei. It is divided into four key phases: prophase, metaphase, anaphase, and telophase. An additional phase called cytokinesis occurs after the complete nuclear division. Before a dividing cell enters mitosis, it undergoes a period of growth and rest. This mitosis. In this module, you will learn about mitosis. Mitosis is a process of cell division that makes possible regeneration of body parts. To elaborate, mitosis is the separation of nuclear chromosomes into two identical daughter nuclei. It is divided into four key phases: prophase,
Oh my god, again.
mitosis. In this module, you will learn about mitosis. Mitosis is a process of cell division that makes possible regeneration of body parts. To elaborate, mitosis is the separation of nuclear chromosomes into two identical daughter nuclei. It is divided into four key phases: prophase, metaphase, anaphase, and telophase. An additional phase called cytokinesis occurs after the complete nuclear division. Before a dividing cell enters mitosis, it undergoes a period of growth and rest. This period is called interphase. In interphase, the genetic material of a cell increases. Prophase. During prophase, the chromatin condenses and becomes short and thick to form chromosomes. Each chromosome has two sister chromatids joined together at the centromere. Mitosis. In this module, you will learn about mitosis. Mitosis is a process of cell division. The chromosome has two sister chromatids joined together at the centromere. The nucleolus disappears and the nuclear membrane starts to break down. The centrosomes, each with a pair of centrioles, move apart to the opposite poles, forming a spindle between them. Metaphase. During metaphase, microtubules of the spindle interact with chromosome. This causes the chromosomes to move and align along the middle of the cell or the equatorial plate. Sister chromatids are held at the centromere, confirming the presence of protein structures called kinetochore at the surface. The kinetochore holds the chromosomes and attaches them to the spindle. Anaphase. During anaphase, the sister chromatids separate at the kinetochores, and the daughter chromosomes, along with the divided centromere, move towards the opposite poles as the microtubule shortens. The poles of the spindle also move apart and help to separate the chromosomes. As the chromosomes separate completely into two sets, one at each pole, anaphase is considered complete. Telophase. During telophase, the chromosomes arrive at the pole and the chromatin starts to decoil. Also, the nuclei reform along with nucleoli. This triggers the reformation of the nuclear membrane. Cytokinesis. Cytokinesis is the partition of the cytoplasm. In animals, a cleavage furrow appears in the plasma membrane, which deepens and divides the cytoplasm into two daughter cells, each containing a nucleus. In plant, new cell wall formation is triggered at the center of the cell by the formation of a cell plate. The cell plate grows outwards till it meets the existing wall, resulting in two daughter cells. In this module, you have learned mitosis is the process of separation of nuclear chromosomes into two identical daughter nuclei. Mitosis involves four key phases: prophase, metaphase, anaphase, and telophase. In prophase, the chromatin condenses and becomes short and thick to form the chromosomes. In metaphase, microtubules of the spindle interact with the chromosomes. In anaphase, the sister chromatids separate out at the centromere and move towards the opposite poles as the microtubule becomes shorter. In telophase, daughter chromosomes arrive at the pole and chromatin starts to decoil, followed by the reformation of nucleoli and nuclei.
I hope it is clear, children.
Yes, miss.
Okay, very good. So now tell me, this chapter is over, all topics we have covered. Now tell me if any doubt you have. See your book, your progress check, review questions. Go through your progress check, review question, and if any doubt you can ask. Then we will move to the next chapter.
Miss, have we done haploid, diploid, and polyploid?
Haploid, diploid, polyploid. Polyploid is not given in your book. Diploid, haploid I have explained. If you have doubt, then I'll explain it again.
Yes, miss, I have doubt in diploid.
Okay. So diploid means, first I'm writing in the chat box, diploid cell, that means a cell having complete set of chromosomes. That you can write 2n, and haploid cell means a cell having half set of chromosomes. See, sorry children, in diploid, inside bracket you write 2n, not n. By mistake it is n. Diploid cell 2n. diploid means total number of chromosome present in a cell. That is called diploid. Okay. Total number may vary according to a species. It may vary from a species to species. So one example is human being. So single cell means one cell of human being consists of 46 number of chromosomes. So that 46 is the total number present in a cell. That is called diploid number. And if it become half during gamete formation, when male and female gametes are formed, this number become half, so become 23. That is called half set of chromosome or haploid. Understood? Is that clear? Who has asked this question?
Yes, miss.
Okay. Polyploidy means, actually polyploidy is not given, but polyploidy means, diploid means what? Di. Di means two set of chromosome, double set of chromosome or complete set. Haploid means half, and polyploid means many. Okay. If more than two number of chromosomes present in a cell, it is called polyploidy. Okay.
Excuse me, miss.
Yes, Srishti.
Miss, if there are 46 chromosomes in a cell, then there will be 23 chromatin fibers inside the nucleus during interphase.
No. Chromatin fibers not in the number, actually it is like network. So if suppose if we are counting the chromatin numbers, so it is equal to chromosome.
Okay, miss, in all cases.
Yes. Only in gametes it is reduced to half. Otherwise it is diploid in number, always diploid in number. Means pair of chromosomes are present. So in a cell, diploid number means complete set, or diploid means two set of chromosome. That means 23 pairs.
Okay, miss.
Okay.
Miss,
Anything else?
Miss, what is kinetochore?
Kinetochore is a proteinaceous structure which is present, means centromere is just constriction. It is a point of constriction, and above the point of constriction means above the centromere, a proteinaceous structure, disc-like structure is present that connect with the spindle fiber. That is called kinetochore. Okay.
So, miss, it helps the chromatin fibers to connect with the spindle fibers.
Yes.
Okay, miss.
So actually spindle fiber attached with the kinetochore, not actually with the centromere, because centromere is just constriction, point of constriction, and this disc-shaped proteinaceous structure associated with the chromatid means associated with the just present above the centromere, and this structure attached with the spindle fiber during cell division.
Miss, why is meiosis referred to as reductional division?
Because chromosome number reduced to half.
Okay, miss. And miss, why?
Using number.
Okay, miss. And miss, why gametes must be produced by meiosis for sexual reproduction?
This I have to explain with the help of a slide. See. Actually, gametes are produced haploid to maintain the normal chromosome number in the offspring. Okay, suppose father is 2n and mother is 2n. See one example here. And this father and mother produce male and female gamete. This produce see, this produce male gamete, which is an in number. And the second, this parent produce female gamete. Okay, so the normal chromosome number of human being is 2n. Am I clear? This is the normal chromosome number of a species, and a species is human being. So normal chromosome number in a cell in human species is 2n. So it should be maintained in the offspring, in the daughter cell, or in the new means in the children. So that is why gametes they fuse together to form 2n, or it maintain the normal chromosome number. The normal chromosome number maintained in the zygote, that is 2n. Understood? Is that clear? So simply you can write offspring gametes are haploid. Suppose it is also 2n. So after fusion it becomes 4n, and 4n means 4 set of chromosome that represent another species. 2n only represent the same species or human being. If it becomes 4n, then it represent another kind of species. So in a single step, new species are formed. To avoid this, gametes are haploid and ensure normal chromosome number of a species.
Okay, miss.
Is that clear?
Yes, miss.
Okay. Anything else, children?
Miss, during cell division, the nucleus is separated. The mitochondria and chloroplast have their own DNA, so they also duplicate and they are divided between the two cells. Miss, but what about the other cell organelles?
Other cell organelles are not divided by own, because they don't have their own DNA and ribosome.
So only one of the two daughter cells receives the organelles.
Mitochondria and chloroplast divide by own, and others are responsible. Other cell organelles are divided, but this is due to action of nucleus. Nucleus is the brain of the cells and controlling center of the cell. So other cell organelles divide due to action of nucleus, but mitochondria and chloroplast have their own DNA and ribosome, so they divide by own.
Okay. In cell division, all the organelles are divided, either by own or with the help of nucleus.
All.
Okay, miss.
Okay.
नहीं पढ़े उठे नहीं।
Any other question? Go through all the questions given in the book. Short answer type, multiple choice, filling the blanks, matching, true, false. Skill type, question, section C, section E. And section E is your homework, must write in your notebook. Rest of the question, rest of the section, section A, B, C, must do in your book. And if in section C, if it is not possible to do in book, then you write in your notebook. Same
Miss
here for
Whole copy.
sorry.
Miss whole E, all the questions of E we will do in copy.
How many questions are there? I think 7 or 6, 7. No. 9
Miss,
questions. 9 questions are there. No, not all. I think some questions you are able to answer in the book. Like in question number 8. Is it a plant cell or animal cell? Give 2 reasons. So if in all the question children, if possible to write in the book, like filling the blanks is given and true false is given. So do in book, otherwise do in your notebook. Like differences, give reason is given. So give reason not possible, so write in the book. So you write in your notebook. Okay. So better you section E, do in your book. No, sorry, in the notebook. Section E in the notebook. Rest of the question in the book. In the long answer type also, what are chromosomes? If you are able to write simple definition in the book, then write it here. Otherwise in the notebook. In short answer type also, state the difference between chromosome and chromatid, centrosome and centromere, aster and spindle fiber, haploid and diploid. This question not possible to write in the book, so do in your notebook. True false in the book, and simple question like what are the rungs of the DNA ladder made of? So this is very simple question, so you can do in the book. I think you don't have any doubt. So today not possible to start new chapter because time up. So next class we will continue with excretory system. Actually, next chapter is genetics, but it is excluded from the syllabus last year, and we are following last year's syllabus. Okay, so we will wait for board instruction, and we will start excretory system first. Children, differences are very important, so must do it. And if any doubt, you can ask in the next class also. And one more thing, children. You have any WhatsApp group with your class teacher? Then add me also.
We have, miss.
So I'm giving my phone number. Add me also, and you can ask in the WhatsApp group also. I have given my phone number. Now you can add me. So shall we wind up this class if you don't have any doubt?
Yes, miss.
Okay. Okay, Shan. Thank you so much, children. Then have you marked your attendance now? Acha sir has already taken the first period. Okay, then thank you so much. Have a good day.